Can you hear me okay? All right. So good morning, everybody. I am Bob Hansaker. I'm going to be talking this morning about structural variation in the human genome. And I don't mind if we make this interactive, so if you have questions, go ahead and wave your hand, and I'll try to notice you, and we can talk about things as we go. So just to sort of level set in terms of definition, structural variation is really the deletion or duplication or rearrangement of large segments of an individual's genome. And over time, the definition of large has actually changed, although as a practical matter today, we sort of set an arbitrary cutoff of 50 base pairs. That's what large means. Things below 50 base pairs are usually things that you'll see registered in dbSNP. dbSNP doesn't like things larger than 50 base pairs, and in the era of sequencing, we've sort of taken 50 base pairs as the cutoff for where we sort of arbitrarily decide things fall into the realm of structural variation. So structural variation really can span the spectrum from something small like 50 base pairs up to whole chromosome aneuploidies. So, for example, the most common aneuploidy in humans is a triploidy of chromosome 21, which leads to Down syndrome, such as illustrated here. Large variants like chromosomal aneuploidies or whole chromosomal arm alterations, they tend to be more deleterious, so they tend not to be inherited. But many smaller variations, as I'm going to show you today, are polymorphic and ubiquitous in the healthy human population. So some of the questions that I want to address today about structural variation is how do we measure it, right? How do we know it's there? How much is there? How much structural variation do we have in the human genome? How does it impact our genomes? And can we associate this variation with common disease phenotypes, and how do we go about that? So I think it's true of all genomics that it's been driven a lot by technology and technological advances, but that's maybe even more true with structural variation. The original look we had at structural variation was really through these kind of banding techniques, as I showed you on that first slide with Down syndrome. And even from these crude microscopy and banding techniques, we could learn a lot, right? This is another picture of a human genome. This genome, if you look at it a little bit, you'll realize is pretty different from the one we just saw, where there was just the triploidy of chromosome 21. This is a cancer genome. So there's been a lot of rearrangements and aneuploidies and sort of shuffling of the genome, which is pretty common in cancer. These microscopy techniques, though, even though they were very early and you could only see things at very high resolution, or not very high resolution, but very large scale resolution, a lot of genetic discoveries were able to be made anyway. This is a picture of a particular genomic alteration here, which is a fusion between chromosome 22 and chromosome 9. It's called the Philadelphia chromosome because it creates a gene fusion that leads to cancer predisposition. And this was discovered through these cited genetic techniques back in the 60s. So there was a lot we could learn even before we could really see genomic alterations at fine scale. The next technology that really drove the field forward was this idea of comparative genomic hybridization, where we figured out that we could stain chromosomes. We could hybridize DNA to chromosomes as a template, and we could stain the DNA. So we could tell excess of DNA by using two-color stains. You could tell an excess of DNA from a depletion of the DNA. And this is similar to looking at the banding, except now we can see much finer resolution, and we can see copy gain and copy loss. And this improved the resolution so that we can see much more gain and losses, especially impactful for looking at tumor versus normal samples and understanding cancer. And then we figured out that we could use microarrays to do the same sort of technique of this differential labeling, but do it at finer scale on arrays. And so as we began to put things on arrays, we began to get the resolution we could see down into this sub-100 kb range. And this is really the time when we realized that these chromosomal alterations were not just something that happened in cancer. They were not just these events that led to birth defects and things. They were really ubiquitous in the human genome, and that healthy people carried a lot of copy number variation too. And still today, array CGH, can be used in the clinic, for diagnostics, and for large alterations and rearrangements, it's still something that we can use today. And then the next really technological development was realizing that we could also get information about copy number variation from SNP microarrays. So SNP microarrays are interrogating the SNPs, but many of the assays are like fluorescent labeling of the two different alleles. But if you analyze not just the sort of state of the SNP, whether it's an AB allele or a BB or an AA, but you actually look at the signal intensity, that signal intensity depends on the amount of DNA. So if you do this across many SNPs, you can look for changes in the level of intensity that indicate gains or losses of whole segments of DNA. So several different algorithms were developed to interrogate the underlying signal on SNP microarrays and ascertain CNVs from that. And that's something that's still used today because arrays are very inexpensive and we have a lot of array data. And so you can still ascertain CNVs from arrays. The resolution you can get depends on the density of the SNP array. And you need the signal across many SNPs because the signal is weak at each individual SNP. Typically you can detect things down, again, sub 100 kb, down to 50 kb or maybe a little bit smaller, and you can detect gains and losses. So with the advent of sequencing, right, we had then a new tool we could begin to look at structural variation. And there's several different ways we use sequencing data to infer the presence of structural variation in the underlying genome. Most sequencing data is actually paired end read. So there's a DNA fragment, and short read sequencing data, you sequence each end of the fragment. So you can look at, and in a typical library, those pairs will be a certain distance apart with some variance in the distance depending on the library. So one of the signals you can look at is whether or not you're seeing read pairs in the correct orientation and at the expected distance apart. If the pairs are too far apart when you map them back to the reference genome or too close together or in the wrong orientation, this can be a signal of an underlying rearrangement in the genome compared to the reference genome. Because when we analyze sequencing data, we usually align those short reads back to the reference genome as the first step. The other thing you can do is just look at DNA abundance. In the same way we can use the SNP array signal to find excess DNA or depletions of DNA in certain regions of the genome, you can do the same thing by just counting the reads as you move across the genome. We think of this as a read depth signal. This is another piece of information we can use. And finally, especially as the reads get longer, you can look to see whether the read actually crosses a place where the genome has rearranged. And as long as the sequence isn't too repetitive, you can map the read there accurately, you can find these breakpoints and this split read signal that again give you evidence of the rearrangement in the underlying genome. So initially a lot of the sequencing data driven by cost was exome sequencing. So you do capture and amplification of certain regions of the genome under coding exons, and from this data, you mostly have then a read depth signal. It's going to be rare that you're going to catch a structural variant that happens to have a breakpoint right in the exon. Maybe you will, but it's more likely that you'll just see a change in the read depth of coverage across several exons that you're capturing. And typically, you can do pretty good calling in the range of like 3 to 5 exons, and there's been a number of methods developed to use this depth of coverage signal to get it at exome sequencing. Exome sequencing is, again, relatively inexpensive, so we have a lot of exome data, and you can analyze copy number variation there. Generally, it has to be large enough. Of course, it has to hit a coding region, and those are kind of the limitations of detecting from exome sequencing. With whole genome sequencing, you can use all those signals I talked about, the split reads and the read pairs and the read depth, and you can combine these signals. A whole bunch of methods have been developed to look at whole genome sequencing data. This happens to be our method that we pioneered in the context of the 1,000 Genomes Project starting in 2011. And ours was one of the first methods to try to integrate many different kinds of signals and to call across many different samples to increase power and and accuracy. And that was one of the things that differentiated our method, but there's many different methods today that work from whole genome sequencing data. Another technique that you can also use to interrogate structural variation is, rather than making sort of a typical sequencing library where we typically have insert sizes, DNA fragment lengths around maybe 400 base pairs, something like that, is you can try to build libraries that have much longer fragments. And the advantage of doing this is you get sort of more physical coverage of the genome. Each fragment is really long, so when you sequence the end of it, you're getting a lot of long-range information. So if you're looking for rearrangements or large inversions, these can be easier to detect from long fragments. So with these long jumping libraries, they're often called, what you get is very long pairwise connectivity. So it's very good for finding these rearrangements. And at the same cost of sequencing, you can get a lot more power to find large, complex rearrangements. The Talkowski lab has been real leaders in this kind of analysis, and they've been very successful in looking at a lot of structural variation in rare disease cases. Another technology that's arrived in the last few years is this linked read sequencing from 10x Genomics. And in a way, sort of similar to jumping libraries, the way 10x works is you prepare libraries with long molecules, hopefully 20 or 10 or 20 kb DNA molecules. They are then, they use a bead technology to actually prepare like a mini library in each bead, where the longer molecule is then sheared up into smaller pieces. And then this library prep goes right into standard Illumina sequencing. And the advantage is when you do it in the bead, you attach a barcode, so that when the reads come out of the Illumina sequencer on the other end, you've got all these short reads, but they all have a little barcode. And that barcode can tell you if the reads came from the same molecule or not. So now you know that a whole bunch of reads come from one region of the genome because they all have that same barcode in them. You pay a little bit of extra cost for the library, and in addition to getting sort of the Illumina information, you get this longer range connectivity and information about whether or not the reads came from the same place, which can help you map into more repetitive regions of the genome. So there have been algorithms written to try to leverage this technology for structural variation. It hasn't taken off in terms of whole genome analysis as much as the 10x technology has taken off for RNA sequencing, but it's still an important and novel way we have to look at structural variation. There's also optical mapping technology. Some of these go way back to the 1990s, and there's more modern ones by Bionanogenomics. So the advantage of the optical mapping is you get really, really long strands of DNA, multi-megabase scale. But then typically you do something like nicking the DNA and attaching fluorophores to it, so you get kind of a pattern of nicks based on restriction sites in the DNA. And you can use these patterns, as shown here, laying down many, many long molecules, and then you can get a very complete picture of the whole chromosome, but at very low resolution because you're only getting these restriction sites that may be 1 kb apart or 3 kb apart or something like that. So these can find very large-scale rearrangements and give you very good connectivity, end-to-end on a chromosome for doing phasing and stuff. So finally, there's probably the transformative technology that we would all love to have, right, is the following. We'd really like a read that's really long, many kilobases. We'd like it to be really inexpensive, and we'd like it to be error-free. And unfortunately, we haven't quite gotten to that technology yet. We've been hoping it's going to arrive for many years. But there are people who have been working on this idea of long-read sequencing. The problem in the past has been both the cost, to some extent the length of the read. You want to get the read as long as possible. The cost has been prohibitively high in the past, and the error rates have been high. And by high, I mean they can be even 10 or 20% base error rates. And this makes the data very difficult to work with. But the technology is constantly improving. And in particular, there have been some pretty exciting announcements lately. I haven't personally worked with this data, but Pacific Biosciences has this new Sequel II instrument. And they've gotten the read lengths up, right? So now you can get often reads that are 100 KB. And one very interesting thing they've done that I think is pretty exciting is that they're using what they call circular consensus sequencing. So if you can sequence a read that's 100 KB long, one thing you could do is try to make libraries with 100 KB DNA molecules, which isn't necessarily easy, right? It's not easy to keep the DNA together at that length scale and sequence it. And you can do that with a PacBio instrument. But the other thing you can do is you can make a library, and then you can do a size restriction to get it down to, say, 8 KB or 10 KB fragments. And then, because the PacBio sequencing, they use this library prep where they put hairpins on the end. When you do that 100 KB read, you actually go round and round the same molecule. And when you do that, you can take, even though the original read has a very high error rate, you can do error correction because you've read the same base maybe 10 times. And so you can correct it and get a much higher base error rate. So you can kind of do this trade-off where you can make smaller reads but with higher accuracy. And so now PacBio, there's a recent preprint on bioRxiv where they're doing this, and they're claiming that they can get their base accuracy up to 99.7, 98.8, something on the order of Illumina reads. The error rate's a little bit higher, especially in homopolymers for indels, but still the error rate's very good. And with an 8 or 10 KB read, you can get into a lot more of the genome than you can with short reads. Also, in their new instrument, they've done some technological advances, so now they can do 8 times as much sequencing on one cartridge. This is driving the price down. So I don't have exact information on what the pricing would be, but you can talk to the platform. But I think we're starting to look at being able to do a genome, under $10,000, maybe well under $10,000. And as the price drops down to this point, you can imagine that maybe we're going to get a lot more long-read sequencing. To date, really, this long-read sequencing has only been done in tens of human genomes, right? Maybe 100 or something like that. There's also Oxford Nanopore, which is another long-read technology. They generate even longer reads than PacBio, but they do have, again, a pretty high error rate. Their products, however, are continuing to get better, and they've announced some new products available this year that should be able to scale up and also reduce the costs of these long reads. So there's competition in the long read space, and maybe it's getting to the point where we're going to start to have larger number of samples being sequenced with long reads. So to sort of sum up this part, how do we measure structural variation? I would say these are the 4 main ways we tend to do it today, right? You can use microarrays, which you can get at really large CNVs, 50 KB sort of range, let's say, and up. The cost is very, very cheap per experiment, so you can do lots of samples. There's exome sequencing, which has limitations that you're only going to see the coding range, and you can only get CNVs, and maybe they have to be between 2 and 5 exons long, the CNVs you can detect, depending on your tolerance for false discovery rate. There's whole genome sequencing, where you can get an awful lot of the genome, although you do miss calls in repetitive parts of the genome. And the short read sequencing has difficulty calling small variants, especially in this 50 to 500 base pair range, because there's just not enough sequence data to get those smaller variants. Longer variants are actually easier to call because you have more data and more information about them. And then there's long read sequencing, which is still relatively expensive, but you get much more complete ascertainment. So that's the ways we look at measuring structural variation today. So the next question I want to talk about is, well, how much is there, right? How much structural variation is there in the human genome? And one thing I'd like you to take away is that there's this qualitative relationship between the number of variants and the size of the variant. So there's many more SNPs in an individual person's genome, many more polymorphisms that are single base pair changes than there are really large CNVs. And this general trend holds across the whole length spectrum. This is a plot from a recent study that's in press at Nature Communications. This was a follow-on to the 1,000 Genomes Project, where 3 trios were analyzed with a whole bunch of technologies, a lot of PacBio data, but also BioNano and Illumina data, and a little bit of Oxford Nanopore at the end, and to try to do complete ascertainment in especially the 3 children from these trios. And what we see here is that we get a spectrum of variation all across the length scale. And it really does fall off. The y-axis, sorry, the x-axis here is in log scale. So you can see that there's this trend that there's many fewer variants as you get out to longer length scales, and it kind of falls off in this natural progression. It's still, I think, a little bit difficult to say exactly how many variants there are in a human genome. But just for ballpark, just some approximate numbers. If you ask how many SNPs there are, there's like 3.5 to 4.5 million. Small indels, maybe 700,000 or 800,000. Structural variants, I don't know, 20 to 25,000. Some reports may say 30,000. Some may say less. And it's very important how you set your cutoff, like 50 base pairs. So I have a question. So I say here for SNPs. SNPs we actually know a lot about, right? We're pretty confident about SNPs. Why do I give this range of 3.5 to 4.5 million? Why am I not more certain? Any idea? Louder? Could be repetitive areas. Other theories? Ancestry. The dominant thing here is just ancestry, right? This is the number of variants as ascertained by the 1,000 genomes in different people, right? And the main effect here, right? That difference between 3.5 and 4.5 million is really just ancestry, right? It depends on whether you're African or non-African, right? So when you go to count variants, ancestry is a really important determination of how many variants you're going to get in each genome. Now, I think counting variants per genome is a pretty good way to look at things, but you do have to worry about certain confounders. The biggest one really is ancestry. You also have to think about, for structural variation, ascertainment at different length scales. Because as I explained, there's a lot more small variants than large ones. So if your ascertainment is not quite as good in like the 100 or 300 base pair range, or conversely, if you're doing very aggressive ascertainment with a little bit higher false discovery rate, you're going to count a lot more variants just by the length scale. There's also different categories of structural variation, as I'm going to talk about, so it kind of depends on what kind of structural variation you're looking for. Your counts may be different if you're measuring different things. And lastly, this is kind of a small but subtle point, there can be different ways to count. So if you imagine, you even take a simple example of a multinucleotide polymorphism, right? So two base pairs that change right next to each other. You could count that as 2 SNPs and 2 changes, or you could count it as 1 dinucleotide change. Well, the same thing applies to structural variation, but on a much larger scale. You can have a couple kb of the genome that's very rearranged, and you can sort of count the edit distance between 2 different forms of that in different ways that give you different numbers of variants. You could count it as 1 big change, or maybe many smaller changes that add up to one thing. Our ascertainment has definitely improved over time. This is an example of like ascertainment from a few years ago in the 1,000 Genomes Project, where we had low-pass whole genome sequencing data, so short reads at low coverage. Multiple methods were used in 1,000 genomes to try to ascertain structural variation, and this is kind of the picture we had. But this picture is not completely right because we didn't have complete ascertainment. If we look today at what we can see from a few genomes done at very high resolution with long read sequencing, we can say that more what the true spectrum of structural variation looks like. This is split into 2 plots here. On the left, this is the small length scale from 50 base pairs to 1 kb. And then on the right, this is 1 kb and up to about 20 kb, and then it's zoomed out. So it looks the same, but you have to imagine that the y-axis is an order of magnitude difference here. So it's really the same curve continuing out. And so you can see that the 1,000 genomes kind of had the right spectrum of variation, except that at the low end, right below like 1,000 base pairs, it's really truncated because we couldn't, with the technology in use at that time, see the structural variation at the small end. The other thing that's important, and I'll talk about this maybe a little bit more later, is that there's these 2 funny peaks that are exceptions to this general trend of having less variation as we get longer. And the first peak over here on the left is around 300 base pairs, and these are mobile elements. These are ALUs. So these are retrotransposons that are copy and paste transposons, and they're active in humans. So a lot of the polymorphism, not a lot of the polymorphism, so one form of polymorphism in the human genome today is that there's these copy and paste retrotransposons that are jumping around our genome, right? And as they jump around, these get detected as 300 base pair polymorphisms, which fall into the range of structural variation. Now, this is always, biologically an insertional mechanism, right? That the transposon copies and inserts in a new region of the genome. But it can be detected either as an insertion, if it's not in the reference, or if the reference genome happens to have a copy of that ALU element in the place it's inserted, then people who don't have that insertion will look like a deletion relative to the reference. And the other spike you can see on the right-hand plot here at about 6 kb, these are L1 line elements, which are also active retrotransposons in humans. A full-length L1 tends to be about 6 kb. And so these two particular biological processes are an exception to this general rule of the relationship between variant size and frequency. So I had just talked a little bit about the ALUs and the L1s. This is kind of the slide, and this shows you the kinds of ALUs and that are active in the human genome. Mobile elements are a little bit of a specialized subfield onto themselves, but it's important to know about them as an important class of structural variation that exists in humans. I also want to highlight that another thing that long reads have revealed is that occasionally there are sort of these complex rearrangements in humans. Sometimes there are parts of the genome that are inverted. It turns out that a lot of the inversions get associated with more complex rearrangements where there's places of the genome that will be duplicated or kind of a little local shuffling of the genome. These are not terribly frequent. There's only like 100 or 200 of them, maybe in a single person's genome that are. That's how many you will see in the genome. It's not that they're mutating in the genome at that rate, but there's just inherited ones that will appear at that frequency. But they do happen. They're much more common, for example, in cancer and in Mendelian diseases. You'll tend to see a higher rate of them. So I do want to talk a little bit about categories of structural variation, mostly because this is often the way we think about structural variation. So if you're going to look at structural variation, it's good to be familiar with the different categories and models that we tend to put things into. Deletions and duplications are the most numerous kinds of structural variation we see in the human genome. So these are this seems to be the predominant way that the genome evolves at this scale, is that you'll just lose a segment of DNA, or you'll have a segment of DNA that duplicates. Usually it duplicates in tandem or nearby. Occasionally a piece of DNA will shuffle and go far away in the genome. And then these will, if they're not under selective pressure, they'll persist in the population and be inherited. There's also, as I mentioned, these more complex inversions and translocations. There's many fewer of these compared to deletions and duplications, but they do happen. There are the mobile elements that I talked about. These have sort of there's an intermediate number of these. There's not as many of these as there are other kinds of deletions and duplications, but there's many more than there would be these inversions or translocations. It's a little bit difficult in some ways to count them because there's very old elements in the genome that maybe are fixed and not polymorphic anymore, but it's hard to know if they're completely fixed in the human population. But maybe there's 1,000 or a couple thousand of this kind of event per human genome. Sometimes we also talk about novel sequence insertions as opposed to duplications. This depends a little bit on the technology we're using to ascertain. If you're using long reads, we tend to just treat insertions as insertions without thinking about whether or not they look like other parts of the genome. When you're doing something that's more alignment-based, it's easier to detect a duplication of the existing sequence as opposed to reads that just don't map anywhere on the human genome. And you can imagine that as we sequence more human genomes, especially with long read technologies, that the reference will be more complete and we won't find these novel sequence insertions, which would really be human sequence that just is not in the reference genome, right? But it's human and maybe exists at high frequency, but it just doesn't look like anything else in the human genome. And the last category I'm going to talk here about is multi-allelic CNVs. I'm going to spend a little bit of time on this just because it's something our lab studies a lot, and so it's something I like a lot, and I want to share with you. So if you think about what I've told you so far, you can have like simple deletions, and this is like what a deletion might look like in IGV in one person. If you looked at that deletion over many people, you might see that it has high frequency in the population. Some people carry it, some people have it in a homozygous state, and some people don't. You can imagine you would have a duplication. It would look similar, but some people have it, some people don't. But then some parts of the genome exhibit a different pattern of variation where there's just a wide variety of copy number, right? There's a segment of the genome, and some people have 2 copies, some people have 6, 8, maybe 20 copies of the same segment of the genome. And we call these multi-allelic CNVs. So we've done some studies of these using 1,000 genomes data. And even from low-pass 1,000 genomes data, you can find thousands of regions in the genome that are multi-allelic. Not all of them are as multi-allelic as these examples here, but there's still thousands of regions like this across the genome. And these multi-allelic CNVs, they contribute a disproportionate amount to gene dosage variation in humans. So in this study we did of the 1,000 genomes data, this was the number of these largish CNVs we were able to ascertain. The blue is showing the deletions, the orange is the biallelic duplications, and the green are the multi-allelic CNVs. And there's less of the multi-allelic CNVs. But then if you ask, well, how many of them overlap genes, right? So that the gene is completely within the CNV. And you see that the ratio begins to shift, right? There aren't many genes that are completely deleted, right? In the human population, that tends to be more deleterious, so it tends not to happen. We'll tolerate the duplication of a gene much more easily than we will a deletion. And if you ask the number of genes affected, the ratio shifts even a little bit more, right? There are more genes that are tolerant to this sort of duplication or multi-allelic effect. And then we can ask another question, which is that if I picked any 2 random people in this room, how many genes are there that those 2 people differ in copy number? One person has 6, one person has 2, right? And the answer is about 75, right? On average, there's about, if I picked me and one other person in the room, there's probably 75 genes where I have a certain number of copies and you have a different number of copies. And what surprised us is that this is driven almost completely by these wildly multi-allelic CNVs. They're the ones that are mostly contributing to gene dosage variation. Duplications in particular, these biallelic duplications, were contributing very little of the gene dosage variation. And this was initially a little surprising, and then we looked at what happens if you stratify by frequency. We asked if these are common variants or rare variants. And it turns out that this effect, where the multi-allelic CNVs are contributing all this gene dosage variation, is pretty much driven by this circle on the bottom, which is the common variation, right? The rare variants, which are contributing some of the gene dosage differences, they're more balanced between duplications and multi-allelic CNVs. And this is because these biallelic duplications, we just don't see very many of them at high frequency. And one hypothesis for why this happens is that we think that perhaps the event that creates this duplication in the first place, where some segment of the genome, we get a second copy of it, is a relatively rare event. But once it happens, it then is easier for incorrect crossover during meiosis to revert that duplication back to a single copy state. Or if the duplicated allele begins to rise in frequency in the population, it will encounter itself. And during meiosis, you can have an incorrect crossover. And you can go from a 2-copy state to a 3-copy state, from 3 to 4. 4 can go back to 3. So it becomes a region of kind of genomic instability as it begins to rise in frequency. So these biallelic duplications tend not to persist at high frequency in the population. They will either die out or they'll become multiallelic. So what's the impact of this variation on human genomes, in particular on our genes and our genetics? Well, I've told you that there's this trend for less frequent, less large variation than small variation. But there's also this other trend that the phenotypic impact per variant by different measures is higher per variant for large variants compared to small ones. So there's a lot of SNPs in the genome, but each SNP, on average, doesn't contribute that much phenotypic variation compared to a large CNV. And I'll show you a couple different examples of this. One is coming back to these multiallelic CNVs. Another thing we did in our study of these was to look at the effect of this on gene expression. So if you change the number of copies of a gene, what happens to the expression level of that gene? It turns out in plants, some plants have the mechanism where they will actually shut down extra copies so that if they acquire extra copies of a gene, it doesn't mess up their biology. But this doesn't seem to happen in humans. In almost all cases, the rule is you have more copies of a gene, you get higher expression of that gene. And the vast majority of genes seem to behave this way. This is looking at RNA expression data from the GeoVetus data set. So we're measuring expression here at the RNA level, not the protein level. So using the GTEx data, there was an analysis done of eQTLs in GTEx, and in particular looking at structural variation. And one of the findings from that was that somewhere on the order of 3 to 7% of eQTLs seemed to be driven by the structural variants that they could ascertain. And this was despite the fact that there's 1,000-fold less structural variations they looked at than SNPs. And this is a pretty consistent number with an earlier analysis done in the 1,000 genomes where they found that structural variation was up to 50-fold enriched for being an eQTL. So the moral of the story here is if you have a structural variation near a gene, there's probably a higher prior on the eQTL being driven by the structural variant than a SNP, if they're both otherwise equally plausible. And another thing that's sort of where the field is right now is beginning to look at really large cohorts, tens of thousands of people, with Illumina whole genome sequencing data and looking at least the structural variation we can analyze and ascertain in this kind of data set. There's a preprint out quite recently on bioRxiv from Ira Hall's lab where they analyzed 17,000 CCTG genomes. And again, they are finding consistent patterns here that structural variation, especially deletions, tend to be depleted in coding regions. They tend to be more depleted when they intersect high PLI genes. Low PLI genes are less affected. And again, the data they're finding is consistent with the fact that a large deletion is more phenotypically impactful than an individual SNP on average. So I want to talk about associating structural variation with common disease phenotypes, whether or not there are any associations, and how we go about looking at this. So when we talk about common disease phenotypes, generally we have to get the sample size up to be able to see an effect. And one of the ways that we can boost sample sizes is through using imputation. So an important initial question is whether or not we can use imputation to impute structural variation into large data sets in the same way we do other variation, like SNPs ascertained from whole genome sequencing data, for example. So imputation of structural variation really depends on the kind of variant. So biallelic deletions, in general, tend to impute fairly well. They impute about as well as biallelic SNPs do. as long as they're well ascertained and in well-behaved regions of the genome so that there are SNPs flanking them, and SNPs we can reliably call that are flanking them, they will impute about as well as biallelic SNPs. Biallelic duplications also impute well in general, or about as well as SNPs. But with the caveat that, like I explained, most biallelic duplications are rare. So given that they're rare, they impute about as well as a rare SNP of the same frequency. For multiallelic CNVs, the imputation results are actually a lot more mixed. It really depends on how many recurrent alleles there are, how multiallelic it is. As something becomes multiallelic, one way you can think about it is that there's many different alleles, each of relatively low frequency. So those alleles tend not to impute as well. Just to give you kind of a qualitative sense of this, I'll show you one example of trying to impute a particular CNV, which is at the haptoglobin-related protein HPR. So HPR helps confer resistance to certain forms of trypanosomiasis. HPR also binds to APOL1, which has been shown to exhibit Africa-specific functional variation, and it's involved in resistance to trypanosomes and also to kidney disease in African Americans. So at the bottom right is sort of a picture of this HPR locus. There is a deletion that you can see in red. This deletion is primarily found in people of East Asian ancestry, and it includes haptoglobin and HPR. And this deletion actually imputes fairly well. There's also this duplication you can see that only affects HPR. This is primarily found in people of African ancestry, and it's kind of odd in that it exhibits what we call this runaway copy number phenotype. Rather than being kind of a uniform distribution of copy number, as you can see at the top, there's a certain amount of people with just 2 copies of this duplication. And then if you don't have 2 copies, you're much more likely to have 6 copies or 5 or 7, right? So you tend to have a very high copy allele or a single copy allele. And if you look at the haplotypes on which these duplication alleles lie, you see the following kind of pattern. This is a plot we call a spider plot. And what this is showing is 280 different haplotypes from people of African ancestry from 1,000 genomes. And the center of the plot is the HPR gene. And then the haplotypes are branching out as they diverge from each other, moving away from the HPR gene. And each horizontal line is a different haplotype. And then these little black tick marks are showing you where there's extra copies of the HPR gene on particular haplotypes. So you can see that most of the extra copies are existing on this one set of haplotypes that are a long shared haplotype. And occasionally there's a few extra copies on other haplotypes, but this is a pattern that would be completely consistent with occasional recombination, right, through these haplotypes. So what this plot suggests is a couple things. First of all, if you wanted to impute this, on that long haplotype, there's going to be tag SNPs that will differentiate a high copy allele from a low copy allele with pretty good accuracy. But if you want to tell between the high copy alleles, like whether it's a 4 allele or a 5 allele, that's going to be very difficult because there just aren't going to be tag SNPs. That's a pretty uniform haplotype. And the 4s and 5s are not differentiated. This is also suggesting that the mutation rate of between 4 copies and 5 copies is happening faster than the SNP background locally at this locus. So imputation here is going to be partially effective depending on what you're going to look at. And this is just kind of like one example to give you kind of a feel for some of the issues you have to think about when you're trying to impute these structural variations. So we know that structural variation definitely has impacts on rare Mendelian disorders, but what have been the stories and the successes in terms of relating it to common disease? So sometimes there are cases where we find CNVs, even simple deletions that seem to have phenotypic effect. An early example was this deletion upstream of the IRGM gene that was found by Steve McCarroll. This deletion polymorphism seemed to hit a promoter of IRGM. It affected gene expression and was associated with Crohn's disease. There's a very interesting, so that's like one example of a way, kind of like with a SNP, you find a variant and it's related to disease with some level of penetrance and kind of this one-to-one relationship. In 2007, there was this really interesting report, originally from Jonathan Sabat's lab, that rather than saying, here's a structural variant and it's related to disease, they showed that there was just an enrichment of large deletions and duplications in people with autism. And it wasn't a particular variant, it was just kind of burden of variation overall in the whole genome. And this was replicated by different labs, and there was a pattern that began to emerge that there were some loci in the genome where there are these recurrent deletions and duplications. So it's not that the deletion and duplication is necessarily inherited, but there are certain parts of the genome where these deletions or duplications would just seem to happen over and over as new mutations with somewhat different breakpoints in many cases, but they would happen at these same loci, and that was a risk factor for autism. But even beyond this number of loci that we've been able to elucidate that have this property, there's still a burden of just excess CNVs around the rest of the genome that's also associated with autism risk. And there's a somewhat similar pattern in schizophrenia, that there's a few loci where these recurrent deletions or duplications are associated with schizophrenia risk at sort of this intermediate penetrance, 10 to 30%. And some evidence for an excess of overall deletions and duplications also conferring risk. Now, in both autism and schizophrenia, the total amount of disease risk explained by these recurrent CNVs is quite small, right? It's not the bulk of the risk, it's a small part of the risk. If you do have an excess of these large CNVs, though, it is a significant risk factor. And this is held up as that we've looked at even larger studies. This was a recent study in schizophrenia where they looked across 21,000 cases and 20,000 controls, looking for large, rare CNVs, 20 kb and up, and they're finding that this excess risk continues to hold. So one way we've tried to also look at, so that's kind of simple deletions and duplications. But what do we do when we get to these more complex, multi-allelic CNVs? So sometimes these multi-allelic CNVs exist, all by themselves in one part of the genome, but actually pretty often they will exist. There will be a locus where there will be several of these multi-allelic CNVs in the same locus. And something we've had pretty good success with, at least in our lab, is deconstructing these loci in the following way. We identify these multi-allelic segments, we make very careful copy number measurements in each one, and then we use either families or statistical phasing to try to determine what are the individual haplotypic copy numbers, as opposed to the diploid copy number we can measure directly. And then given that we have these different segments and these haplotypic copy numbers and they're all phased, we can begin to relate that to what we think are the underlying structures and alleles that are going on at the particular locus. And often we find that there's a lot of complexity at these loci. This was an early example that Linda Betker worked on in Steve's lab. This is the MAP tau locus at chromosome 17q. And at this locus in the genome, which has been studied by many different labs, there's a 1 megabase inversion that's fairly common, mostly in Europeans. So there's a megabase in the genome that's actually flipped. And near the edges of this inversion, there are also these copy number polymorphisms. The top is showing you, I believe it's the HG19 reference, and it's showing you these 4 blocks in different colors. And then below, this is showing you pictures of the different structures we see occurring at high frequency in humans. Some in the forward, with the inversion in the forward orientation, those are the top 6 labeled H1, and some with the inversion in the reverse orientation. And then you get these polymorphic copy number changes. And Linda found that 2 of these haplotypes in particular have gone to very high frequency, mostly in Europeans. And Both of them, as it turned out, created fusion genes involving this gene called cancel-1. And this is a loci that's been associated with several different phenotypes, including fertility phenotypes. A similar study was done at the amylase locus. Amylase is very polymorphic. Some people have up to 20 copies of the amylase 1 gene, and there's a bunch of genes altogether at the amylase locus that vary in copy number. There's a lot of complex structures at the amylase locus. They vary in copy number. Sometimes the copies are inverted, as shown by these little arrows. But you can do this kind of deconstruction of the different haplotypic structures and then look at their frequency in the population. By doing very careful measurements and analysis here, Christina Usher was able to show that some previous reports of a strong association to BMI at this locus were really due to misunderstanding the structure of the locus and not measuring the alleles accurately. And as a third example, in probably one of the most important results recently in psychiatric genetics, was fine mapping a schizophrenia association. If you look at the Manhattan plot that's in the center here, the strongest schizophrenia association is this big peak, sort of in the middle. That's at the MHC locus on chromosome 6. It's notoriously difficult to fine map things at the MHC locus, but Ashwin Sekar, who is an MD PhD student in Steve McCarroll's lab, fine mapped this association signal, and he fine mapped it to this gene called C4. C4 is also one of these complicated multi-allelic loci. To understand C4, you have to understand that there's actually two forms of the C4 gene. They're called C4a and C4b. They differ at 4 amino acids at a specific binding site in C4, so they have different biological activity. There's also what we call long and short forms of C4, and these are distinguished by whether or not there's this large deletion that exists in intron 9 of C4. And so you've got these 4 different forms of C4. You have a C4a that's long or short, and a C4b that's long or short. And then the whole thing is copy number variable. People, at least the people I have looked at, have between 2 and 8 copies of C4. And there's a whole mixture of whether these copies are C4a or b or long or short. It exists in many different structural haplotypes. What's shown here is the ones that Ashwin ascertained that are at relatively high frequency in the population. We now know that there's 20 or more different structural forms of C4 that are at fairly high frequency in the human population. And Ashwin showed that this association signal to schizophrenia localized to this region, and in particular to the number of copies of C4a you have, more or less independent of whether they're long or short. And part of the lesson of what Ashwin did here and what he learned was that he used this idea that there's these multiple recurrent forms in a very important way. So these multiple recurrent forms can be either sort of a blessing or a curse, right? Because these forms are recurrent, they're difficult to impute and they all exist at low frequency. So what's being shown on the left here, for example, the second category, this is what we would call an ALBS haplotype. So there's a haplotype with 1 long copy of C4a and 1 short copy of C4b. But what those little barcodes on the side are showing, these are the SNP haplotypes that this ALBS form resides on. And you can actually see that there's completely different SNP haplotypes that have the same C4 structure on them. So this suggests that this structure is recurrent. It's occurred through multiple mutation paths that lead you back to the same structure. But Ashwin showed on the right that the association is to the structure, independent of which SNP haplotype it exists on. So this recurrence makes it difficult to analyze the locus, but you can also use that to show that it's much more likely that the association is to the structure as opposed to the background SNP haplotype. So one final note, I also wanted to talk a little bit about a not inherited structural variation, but somatic structural variation. And there's been a lot of new work recently in looking at clonal hematopoiesis. So this is mutations that arise in your hematopoietic stem cells that produce blood cells. You can see these in the blood as sort of clonal populations of blood cells that arise. And so you'll see a fraction of the blood cells that are carrying new variants. Recent work has shown that this increases sharply with age, at least in some people. So there's a dramatic increase of this kind of somatic mutation with age. It can be a signature of different kinds of blood cancers, and it can even show up quite early, pre-diagnosis. And some recent reports have indicated that there can be other phenotypes associated with this, such as increased risk for coronary artery disease. And there's been some recent work associated with Broad. Po Rouleau at Brigham and Women's and Giulio Genovese here at Broad developed a new method called MOCA for detecting these somatic CNVs. They use phasing to really increase the power for looking at these somatic events. And by using phasing, they're able to detect these events at extremely low fraction, down to as low as 1% clonality in blood. They did an analysis of this in the UK Biobank that was published last year, and they found thousands of these clonal events, and they were even able to show, in several cases, germline variants that were conferring predisposition to this kind of clonal somatic mutation. So this is like a different kind of structural variation that's happening within our bodies that may have a lot of importance for certain phenotypes. So to sort of wrap up and talk about the main messages. So there are fewer structural variations overall in our genomes than there are SNPs, but there's still a very large phenotypic impact from structural variations. Maybe 3 to 7% of EQTLs, and there's on average more impact per variant than for SNPs. Long-read technologies are really helping to reveal the full spectrum of structural variation, especially at some places that have been difficult in the past to ascertain with other technologies. It can be difficult to analyze these structural variations because sometimes they may have faster mutation rates than nearby SNPs. They may not be well tagged, therefore, and they're hard to impute. And sometimes it's very important to dig into the underlying structure and figure out what it is about the structure that can be related to phenotype. Imputation into large cohorts is often needed to be able to test for associations, as well as really understanding what the structures are and maybe testing different features of the structures to figure out the correct association. So with that, I have probably a couple minutes for questions before we move on to the 9:30 talk. And I want to thank a lot of people that gave me background material and have collaborated with me and otherwise helped with this talk. Thanks very much.